Hello and welcome back to Communicable, the podcast brought to you by CMI Communications, ESCMID's open access journal covering infectious diseases and clinical microbiology. My name is Angela Huttner. I'm an infectious disease doctor at the Geneva University Hospital in Switzerland and editor-in-chief of CMI Comms. I'm joined today by my co-host, Erin McCreary, ID pharmacist and Director of Infectious Diseases Improvement and Clinical Research Innovation at the University of Pittsburgh Medical Center and editor at CMI Comms.
Hey, Angela, thanks for having me. I'm very excited to be here for this conversation. These are some of my favorite humans that I would wake up at 4:30 in the morning for to record this. So very excited for this conversation.
Yes, thank you, Erin. It is 5 o'clock in the morning now, where you are.
5:19. Let's go. Yeah.
I know. But this podcast, you will find out, is being recorded on several continents right now. But just one last note on Erin. Erin is also co-founder of Breakpoints, which is a groundbreaking podcast belonging to the Society of Infectious Diseases Pharmacists, SIDP. I've been a fan of their podcast for years, and I continue to be inspired by it. Breakpoints will be celebrating its 100th episode in July, so make sure to look out for it. This special episode will focus on all things tetracyclines. It'll be reviewing the 6 drugs in the class, comparing their PK/PD and their roles in therapy. It should be a really great listen, and it's dropping on July 26th. Back here on Communicable, we today will be unpacking BLING-3, the trial that included 7,000 patients to finally answer the question of whether giving beta-lactams by continuous infusion rather than standard intermittent dosing decreases mortality. Very hard endpoint. We have as our guests Professor David Paterson, who is in Singapore right now, and Professor Josh Davis in Newcastle, Australia. These are the 2 ID doctors who were investigators on this trial, which was otherwise driven mainly by intensivists.
Yeah, and we're so excited for this conversation. So I'll introduce David first. So David Paterson is a professor of medicine at the National University of Singapore. He's also an honorary professor at the University of Queensland. Professor Paterson directs Advance ID, which is a clinical trials network comprising more than 40 hospitals across Asia, and it's funded jointly by the Wellcome Trust and several Singaporean institutions with an aim to conduct clinically important trials in the field of AMR. And then on a more personal note, David and I also have an invisible string connecting us, wherein he was actually at the University of Pittsburgh just prior to when I arrived there, early 2000s, but was instrumental in building the first stewardship program, and many of his principles still exist there. So David, is very excited to see you today. Great to talk with you, and welcome to Communicable.
Thanks so much, Erin. Great to be here.
And Josh Davis, as a BLING-3 investigator, you are a guest today, not a co-host. Josh is professor of infectious diseases in Newcastle, Australia, and he's an investigator on many ID clinical trials. He's also editor at CMI Comms and therefore often a co-host.
Yeah, hi. Thanks for inviting me. It's nice to be on this side of the desk.
So, thanks to Josh, we now start these episodes with a get to know you question. Our question today: what is a favorite memory you have from residency? Erin, you first. Hosts first.
Am I first because I'm the closest to residency? Residency was I loved residency. I'd go back tomorrow and do another specialty just to it was like I know Angela's making a face, but it's the only time in your life where I know you make like $2 an hour, but you get paid to learn, which is amazing, right? Enjoy it, folks. Trainees out there listening, I know you're struggling, but it's the only time in your life when you get paid to learn, and then it's, then all bets are off. But I will say, so for those Grey's Anatomy fans out there, I know this is a global enterprise here, but I feel like after 20-something seasons, maybe people have heard of Grey's Anatomy, but in that medical show, all the residents had Joe's Bar, which was right across the street from the hospital, and they hung out there every night. And I went to the University of Wisconsin, and there was truly the same situation. So there's a restaurant right across the street from the hospital, and they had the best French fries in the whole wide world. I dream about these French fries. And when I left Wisconsin, people jokingly said that this restaurant was called Oliver's. They're like, Erin, Oliver's is going to close when you leave because you and your friends are there eating French fries every night. And we joked about this. I kid you not, that restaurant closed 6 months after I moved out of Madison. And I was like, oh no, we were keeping it in business.
David, you're up next.
Yeah, this is a patient, and he actually led to the first publication I ever had. And he was an old guy with peripheral neuropathy in both feet and an ulcer and an infected ulcer on his foot. I said to him, how long have you had diabetes? And he said, everyone asked me that, and I've never had diabetes. He had an interesting sort of personality, and to cut a long story short, it turned out that he had had leprosy, and he'd spent 20 years in what was called a leper colony. In the 1930s, he went off to an island because he was diagnosed with leprosy and spent 20 years, so until the 1950s when they were closed. And it was really interesting. The stigma attached to that diagnosis was very, very, I guess, similar, although different, to, the stigma at that time of people when I was doing my residency, meaning that time, who had HIV. And, at that time that was incurable. Sometimes families disowned people. So really, really interesting to have met someone in that situation. I won't go into details about the manuscript that came out of this, but he learned to play the harmonica. There's not much to do when you're on an island because you've got leprosy. And he had these huge varices under his tongue. And this was from playing the harmonica all the time. And he had this massive hemoptysis, which I entitled Harmonica Player's Hemoptysis. And that was my first publication. So a really memorable patient encounter, but somehow got a paper out of it as well.
Wow, well, that's so interesting. Thanks for sharing that.
Yeah, and I don't think nowadays we run into very many people who have had to live in a leper colony.
No way.
No, I'm glad I went first because my story is so much worse than that story. That was awesome. Josh, I feel bad for you, but why don't you share your memory?
Yeah, hard to
It's hard to follow. Yeah.
First, I've got to agree with you, Erin, about going back and doing training again. I went to an information evening last week for all the different ID training programs around New South Wales. Basically, they were kind of pitching their programs to prospective trainees, and I was there pitching my one. And when it was my turn, after having listened to all the others, man, I want to go back and do it again, like at all of your hospitals. It sounds really enticing.
You guys are
and
so
training
good.
is fun. Yeah, but that's not my memory. So before I was in the infectious diseases training program, I was ID curious, looking around at different specialties. And I was rounding at the time with an ID physician who was about to retire, who was months away from retirement. I can't even remember what patient we had just seen. We saw a patient and he just suddenly said out of the blue, you know what? Infectious diseases is really interesting. I really like it. And I reflected, I've never heard a specialist who's about to retire say that. They usually say, I'm really into photography now. My golf game is getting really good. They're normally burnt out and a bit sick of the thing that they've been doing for 50 years. But that just made me think, yeah, actually, ID is a good specialty. So, and I've thought about that often since, and I still would agree with him, I think.
And I think, Josh, you'll be saying the same thing, probably, it sounds like, if you're actually going around the country and telling people you wish you could redo residency. So I think you'll be just as inspiring. For my part, I thought about this a lot. The thing I kept coming back to was not necessarily any one patient encounter, although there were some very memorable ones. So I trained in New York City at Columbia Presbyterian. And for whatever reason, in the ICUs, both the medical and the cardiac ICUs, all the nurses were Filipino. So I have this memory of these amazing, and they were all women, these amazing Filipino women nurses who just took really good care of the patients. But when I think back, I realized that they were also taking very good care of us. We were very young. I was a second-year resident, and I was in charge of that whole ICU every fourth night, all night. Me and an intern who had, even less experience than I did, and we thought we were running that place. On paper, we were running that place. But I think, looking back, it was the nurses who were they were silently coaching us. They had so much experience. They had so much knowledge. But these women managed to do it in such a way that they respected our illusion that we were the ones in charge. They were just kind, and this wasn't always the case. As we rotated each month to different units, there was hazing of the young doctors on other units. There was sometimes, tension, these young doctors coming in and giving orders. But these nurses, they just had such grace, and they were very cool. They took good care of us.
All right. We have a lot of questions about BLING-3, so let's go ahead and dive in. For listeners who may or may not have been following the BLING saga over the last several years, here is some context. So the BLING studies evaluate the use of beta-lactams at the highest level. Beta-lactam antibiotics are time-dependent antibiotics, which is very fascinating. There are very, very few drugs that fall in this domain. And some would say that beta-lactams are really the only class that truly exhibit this, and every other drug is exposure or AUC-based, but there are a few other time-dependent. And when we say time-dependent, it means they kill bacteria better when their concentrations are kept at a consistent level, and that level is something above the minimum inhibitory concentration of the organism. And so there isn't really any additional benefit to a concentration-dependent kill, wherein you get this high peak or you achieve a certain high concentration, and that would then enhance the kill. It's just staying steadily over the MIC. So that's pretty unique. And in that, then, it would make sense that if you keep your blood concentrations at a time-dependent, steady antibiotic concentration continuously, rather than having these peaks and troughs with these short half-lives, you perhaps would optimize the kill of the organism in your patient. So hence the rationale to administer a beta-lactam antibiotic via a continuous infusion, wherein you're taking the total daily dose, putting it in one bag, and running it over 24 hours, rather than giving it every 8 or 6 or even 4 hours like we do for some. On the pharmacokinetic side, studies have shown a clear benefit in when you reach your PK targets with continuous infusions, you see more kill and modeling and et cetera. But then when we translate that to clinical trials, there hasn't really been conclusive evidence of microcure or improved clinical outcomes when you're optimizing these PK/PD principles. And then, of course, showing the hard outcome of a mortality benefit is the holy grail of any study, right? Particularly in infectious diseases. So the BLING trialists have sought over several years and 3 trials now to study this. And so today we're talking mostly about BLING-3, which was just published in JAMA. But Josh, because history is our greatest lesson, can you give us a brief summary of BLING-1? And then David, gonna come to you to summarize BLING-2 to get us up to speed before we talk about 3. It's like the Fast and the Furious movies, I feel like. They're all so good, but you gotta work your way through them.
The difference is the sequel gets sequentially better in the BLING trials rather than sequentially worse like in most movies.
Thank you. Okay, yeah. Fast and Furious 2 and 3 are so bad. The first one is golden, and people will argue this forever. Are there going to be 9 BLING trials? One can only hope. Okay, BLING-1.
Yeah, so BLING-1 was really a pilot trial, and the BLING-1, and in fact the whole BLING series, was really the idea and the baby of Jeff Lipman, who's an intensivist from Brisbane that David used to work with clinically, and David and I have worked with on a research level. And so Jeff had done a lot of this in vitro and observational kind of work and wanted to translate it into a clinical trial. BLING-1 was a pilot trial, like I said, where 60 people were randomized, so 30 in each group, across 5 Australian intensive care units, to either get continuous infusion of beta-lactam antibiotics that were used in the ICU. So that was primarily piperacillin, tazobactam, or meropenem. So continuous infusion or conventional intermittent dosing. And the endpoint was the proportion of patients in whom a random level taken, a sort of steady state level taken on day 3 was above the MIC of an average organism. In other words, it was a PK endpoint because it was a proof of concept trial, pilot trial. And that was achieved in 82% in the continuous infusion group, 30% in the intermittent dosing group. Kind of a self-fulfilling prophecy, but really the idea of a pilot trial is to show that you can do it and then convince a funding agency to fund a bigger trial. The other thing in BLING-1 was that clinical cure was 70% in the continuous infusion versus 40% in the traditional dosing group. So interesting there, but very small numbers. But that convinced the Australian NHMRC to fund a larger trial, which was BLING-2. So over to you, David, for that.
Yeah, I'm going to diverge back to Pittsburgh for a second before I get on to BLING-2. There's a famous Australian intensivist called Rinaldo Bellomo, and he spent some time in Pittsburgh. And he said to me, when we met in Pittsburgh, clinicians have got to do clinical trials. And at that time, I had a wet lab and also did molecular epidemiology. But it was so important what he said to me, because when I got back to Australia, I think Josh had the same mindset. And together we sort of set up a clinical research network modeled on what Ronaldo had done in the ICU network. And that's what led to the Merino trial, the camera trials, the SNAP trial, etc., etc. But the other thing that Ronaldo said to me is do things in a stepwise fashion. Do a small proof of principle pilot. Then do a study of 300 or 400 patients to really get a signal that what you're studying works. And then you can go to the funders and say, look, it's going to require 5,000 plus patients. We need $10 million. Sorry, Erin, this is what it costs to do a trial in Australia, not in America.
I was going to say $10 million.
Yeah,
You're going to 2 patients in America for that.
And that was Ronaldo's philosophy. And it's the one that the ICU Clinical Trials Group in Australia have used, and was exactly the philosophy followed that led to BLING-2. So BLING-2 was in 25 ICUs. It was 400 patients. And the same philosophy, let's try out. We randomize patients to either continuous infusion or a 30-minute intermittent infusion. And this was still in the time when people were using Timentin. So it was Timentin or Piptazo or meropenem, either continuous infusion or 30-minute infusion. The endpoints were things like ICU-free days, secondarily 90-day all-cause mortality, clinical cure. And while there were some trends in terms of ICU-free days and clinical cure, there was really no difference in 90-day all-cause mortality. And I remember after the trial was published and there was discussion, should we go and do BLING-3? I actually said no. I said, look, there was no signal that this improved mortality, and mortality is what matters. But luckily, and I think we know this from our clinical experience, sometimes the ICU docs are a bit more forceful than the meek and mild ID docs or clinical pharmacists. And the ICU people said, no, we're going to go for it. Really credit to Jeff Lippman. Jeff turned 70 a couple of years leading up to BLING-3 when he was maybe 65 or something like that. This is the culmination of his career. When he got funding to do BLING-3, this was it. This was Jeff's hypothesis, which had been his hypothesis for 20 years. Was it going to pan out in a big, big trial? So all credit to him that BLING-1, BLING-2, now getting funding to go ahead and do BLING-3.
Just want to push back a little bit, David, on one thing. I agreed with after BLING-2, I found the results quite disappointing, but there was no statistically significant difference in mortality, but there was an absolute 2.5% benefit in mortality in the continuous infusion group, which was nowhere near statistically significant. Could have been a chance finding, but could have been a real finding. And that was the kind of question for BLING-3. Is there really a 2.5% mortality benefit, or was that just a random finding?
And that's so important, isn't it? Just in terms of sample size when we're doing clinical trials, to call things off and say, look, didn't reach statistical significance. That's it. Answer, the question's answered. I think increasingly we realize that you've got to do big, big, big trials to really show whether stuff is just a random chance, or whether it's the real deal and there is a true difference between therapies.
Just David, coming back to the discussion of Jeff Lippman, it's funny because I was just emailing with him yesterday because he's very active as an editor at JACC, and he was asking me to review. And of course, I congratulated him on BLING-3. And it's interesting what he responded. He said, oh, yeah, I'm moving along and I've given Jason Roberts and Joe Dulhunty, they had to take over most of that BLING stuff, which, by the way, took me 30 years to prove. So I thought that was really sweet. We will be discussing what does BLING-3 tell us, but he's clearly in the camp of a very positive study. But
yes, that's right. Jeff was at medical school with Nick Daneman's dad in South Africa. And like now Nick has done Balance and is starting Balance Plus. It's like
full circle.
All these threads,
Yeah. Invisible strings, Erin.
Exactly.
Not so invisible.
Indeed.
So,
all right.
Erin, you take this one.
BLING-3, should we? I know, we're all built up for it. So BLING-3. So I'm excited to talk about this too, because BLING-2, as an avid fan of PKPD, obviously been following the trials, and so I think the other big thing that stood out to me with BLING-2 is that 19% of the patients had a documented infection. So it's like, well, it's hard to tell the impact of antibiotics when only 1 in 5 are slam dunk infected. So that needle shifts a little bit in BLING-3, and we'll talk about some of the details. But first, before we get there, Josh, can you explain to us the overarching design of the trial?
BLING-3 went multinational, much larger, and needed funding from multiple different countries to get the trial done. The sample size estimation was based on that observed 2.5% mortality benefit in BLING-2. And so the sample size we were aiming for was what actually was a bit smaller than ended up being recruited, which was about 7,000 patients. There's a reason it was a bit larger, which is about a PK/PD sub-study. But anyway, 7,000 patients were randomized to either continuous infusion or intermittent dosing of piperacillin, tazobactam, or meropenem. And to get into the trial, the patient had to be in an intensive care unit. Clinician had to have decided to treat them with one of those 2 antibiotics, and they had to be critically ill, which there was a list of possible diagnoses and condition that was fairly pragmatic. So as Erin said, some of these patients turned out to not have an infection. But I would say that if you look at most sepsis epidemiology kind of trials, only about 50% maximum of people with sepsis have an identified pathogen. Most of them truly do have sepsis, particularly the pneumonia one. And the top line finding anyway was that mortality was numerically lower. The 90-day mortality was numerically lower in the continuous infusion group at 24.9%. compared with 26.8% in the intermittent dosing. The devil is in the detail here a little bit. The primary analysis, prespecified analysis, found that the p-value for this comparison was not quite statistically significant. The 95% confidence interval ranged from an absolute benefit of 4.9% to an increase in mortality of 1.1%. However, a pre-specified secondary analysis of that primary endpoint was an adjusted analysis, which is traditionally what's done in most large randomized trials that adjusts for any baseline differences in important prognostic variables. And that adjusted analysis, there was a statistically significant benefit. So that's the primary outcome. There were also some important secondary outcomes such as clinical cure. Clinical cure was also statistically significantly higher in the continuous infusion group.
But Josh, remember you said to everyone, so just going back, we had the trial steering committee. First of all, had to sign agreements that we wouldn't spill the beans of the results. And then once everyone had signed that they wouldn't spill the beans, we had the results reveal. And Josh had the greatest idea, and he said, stop. Before you present the results, why don't we all vote on whether there was going to be a statistically significant mortality advantage or not? And I know that I voted and said, no, there would not be an advantage. And I think, Josh, you were also in the no camp.
Yes.
I can't remember what the ICU people, how they voted. And then they had this, we saw that P equals 0.08. And I thought, Josh and I sort of won, but we're not that comfortable that we won. And then they did the adjusted analysis. And I was sort of on the hand up. Like, did you just make up this adjusted analysis after you saw P equals 0.08 and fiddle with a few things? But no, it was pre-specified in the statistical analysis plan. So before they saw any data at all, they had pre-specified that they would adjust for, I think, Apache score, whether you'd come to ICU from elective surgery, and whether you had meropenem or pip/tazo. So this was all completely pre-specified, and that adjusted analysis it did come out statistically significant. So certainly I thought, well, I voted wrongly. It did actually have a positive result. And I mean, Josh, is that your take as well?
Yeah, I think so. I mean, one could argue that adjusted analysis should have been our pre-specified primary initial analysis, because most clinical trial statisticians say that is the proper way of doing things. And I think the reason, I remember having discussions, and I think the reason we didn't is that the people we were working with, the George Institute and ANZIC CTG, they're very conservative. They like making sure things are done exactly correctly, and they always sort of take the more conservative side of things to make sure findings are true, kind of thing. But I think that was probably overly conservative, not using the adjusted as the primary. And the conversation would probably be a little bit different now if we had.
So I think here I will just quickly recap for the listeners, because you two were deep into this study. Obviously, you were planners of this study and you know it. What's happening here is that these 7,000 patients were included, and you looked for mortality at 3 months after randomization, and you found that there was less mortality in patients who got continuous infusion. It was 1.9% absolute difference, and that just missed statistical significance with a p-value of 0.08. We'll go quickly to the p-values, even if maybe we like confidence intervals a little better. But in these adjusted analyses that you're talking about, the mortality difference, the absolute difference was 2.2%. And that was clearly significant, statistically speaking. I'm just recapping for those who don't have this familiarity, because this is, of course, where the money is, right?
Can you guys talk a little bit more about why throughout the BLING studies, a 90-day mortality endpoint was chosen as the primary outcome? Because I think this is a conversation we have wherein for an acute infection, is this the appropriate endpoint? A lot can happen in 3 months, as we see in the hospital, and you may survive your initial sepsis encounter. And I know this came up with Marino and other studies as well. So enlighten our audience a little bit about why this was chosen throughout these studies.
Do you want to go first, David? I've got a view on this, but you probably have been in it too a bit more with Marino.
Yeah, so, in our most recent game changer trial, it was 14-day all-cause mortality, because I think we thought, like you, Erin, if we're talking about antibiotics and an acute serious infection, we don't want to be capturing the people who died of their underlying disease. I have to say, when I first heard the ICU people talking about 90-day all-cause mortality, I was saying, no, you're making sure that this is going to be non-inferiority, not superiority. But then I saw this paper by Davis et al. About mortality over time in patients who had sepsis. And
is that
maybe
this
Josh,
guy? This Davis?
Yeah, this
Josh.
J.S. Davis. So
who's
Josh,
this guy talking about mortality?
So this was something I did as part of my PhD, and we took a cohort of patients hospitalized with sepsis, around 1,000 people, and followed up over time, and then used a technique called relative survival analysis, which compares them to background population life tables and tries to estimate the excess mortality that's attributable to the sepsis. And the bottom line is these people died at an excess rate than the background population on a survival curve for about 2 years. The curve didn't start plateauing until after about 2 years following their sepsis episode. That's not a unique finding. That a lot of studies have shown that. One can argue why, but the other observation that people have made is that after an acute episode of sepsis, the risk of having a myocardial infarction, of having a stroke, of having any major cardiovascular event is hugely increased for several months after the episode. Endothelial dysfunction increases and takes months to settle. The argument that people dying 3 months after an infection is probably due to their underlying comorbidity is a specious argument in my mind. Just because something is not directly visibly related to the infectious insult, it probably is invisibly related to the infectious insult.
Yeah. Thank you. That's super helpful. So in the key point section, in the final meaning punchline of the trial, they say, in critically ill patients with sepsis, continuous versus intermittent beta-lactam antibiotic infusions did not significantly reduce 90-day mortality. So that's if you're only reading the box, which is why we don't just read abstracts. It has somewhat of a negative frame of, there's no difference in the study. But you guys published a corollary meta-analyses with this trial and all others to date, which also shows a statistically significant reduction in mortality with continuous infusion when you look at this meta-analysis, which I think included 17 randomized trials. So there's that. And then we talked about this a little bit, but let's get into it in a little more detail. So the secondary outcome then of clinical cure by day 14, so looking at that more acute endpoint, was actually was statistically significantly better. Can you talk a little bit more about this clinical cure? So how did you define that?
Clinical cure in BLING-3 was defined as the completion of the beta-lactam antibiotic course by day 14 and without recommencement of antibiotics within 48 hours of cessation for the same infectious episode.
Someone needs to play devil's advocate. It's going to have to be me. Clinical cure. I agree that the results on paper, they're significant. There's definitely a difference, and that does pull the conclusion toward a very positive study. But again, this is an open label trial, and that outcomes, it's subjective because the definition of it seems pretty solid, but it's ultimately defined by a doctor's choice to restart or prolong antibiotic therapy, right? And that doctor knew what the patient was getting. So that doctor will have biases. What do you say to the fact that, this will always be an open door for anybody who wants to criticize this trial and say, no, I cannot believe this clinical cure because, it's a subjective outcome with an open label intervention. Josh.
That is a subjective endpoint. Clinicians are aware of what the treatment allocation of patients were. To me, I think I wouldn't put too much weight on their clinical cure data because it's subjective. I think really what we should be focusing on is the 90-day mortality data, and that's clearly very objective. One thing it's important to point out is in all the pre-specified subgroups within the BLING-3 trial, the 90-day mortality was in the same direction, as in the direction of benefit. There were no subgroups in whom it was no effect at all or harm. And then importantly, Erin raised the meta-analysis that was published in the same journal, and it had really quite strong statistical support for mortality reduction with beta-lactam infusion with odds ratio of 0.86. And what's known as a Bayesian posterior probability of 99.1% that continuous infusion reduces mortality. And just as a slight tangent, why was BLING-3 not placebo-controlled is not feasible to do in such a large number of patients at such a large number of sites for such a small budget. So that was one of the kind of feasibility questions in BLING-1 is, can we do a trial like this in a blinded way with a placebo? And it's possible to do, but it's very resource intensive. Do you have anything to add, David, about that clinical cure endpoint?
No, I think you're exactly spot on. The whole flavor of the trial being so pragmatic. I remember we had arguments in the trial steering committee that were shut down by the ICU people who've got immense experience about doing this trial, these sort of big trials, is they wanted to get people in the trial. To get thousands of patients, you've got to be so pragmatic, you've got to have very few exclusion criteria. You really also have to be very careful about what data you collect. Endpoints have also got to be quite pragmatic. The overarching thing is it's not the primary endpoint. The primary endpoint is all-cause mortality. Clinical cure is a secondary endpoint. Now, then people might say, oh, look, the p-value didn't get there. But the really neat thing that the JAMA editors did, and also the whole BLING-3 team did, was to simultaneously come out with a systematic review and meta-analysis. BLING-3 is now being looked not just in isolation, but in the totality of the world's literature. The Mercy trial, the little bits and pieces, 100-patient trials from around the world, everything that's been done on this topic put together. And one thing that really impressed me was that they graded in the systematic review, they graded the sort of quality of evidence, and the 10 trials out of the 17 that had a low risk of bias contributed like 95% of the data. If we've got to make a decision today on what we're going to do about our patients tonight or later today that are seen, the evidence has stacked up. I don't think there's ever going to be a BLING-4. As I mentioned,
50,000
answering
patients.
all our questions.
I mean, I mean,
like,
what would you do? I mean,
I think it's answered, and maybe if you've got, it's interesting as now a trialist, and I don't know if you've seen this, Angela and Josh, people on X or around the world can throw mud at you with your trial results, don't agree with their preconceived notions. You can find a million little methodological issues in trials. We're not talking here about industry trials, $100 million, everything to the nth degree. This is a pragmatic trial trying to answer a clinical question, and I think the question is answered.
Yeah. And honestly, the people that enroll in to the nth degree trials are not the patients, right? They're not the patients you're seeing in your ICUs. I think this is the right way to do, particularly this patient population. None of these patients are gonna fit criteria. I think at this point, the body of evidence is substantial. Even I'm thinking back even to flashing back to residency, right? So I started my residency in 2015. So in 2014, I remember there was a meta-analysis out of the Chicago group, Jim Rhodes and his team, that looked at piptazo and intermittent, so 30-minute infusions to prolonged or extended, so 3 to 4 hour infusions, not continuous, but that body of literature, which we haven't really talked about, but same concept. And that was a meta-analysis of everything from the little observational to the RCTs, and they found a mortality benefit. And then it's like they knew the haters were going to hate. And so then they did another saying, okay, here's the meta-analysis of all data in totality, but then here's only the RCTs. And even when you look at only the RCTs, there's still a mortality benefit. And that was 10 years ago. So I think at this point, we have our answer. And congratulations to both of you guys on just a tremendous trial.
Yeah, and I'm
the haters hate, you just have to shake it off.
Exactly.
I would have one remaining question for both of our guests. It's a shift in culture, it's a shift in practice for a lot of hospitals to go from intermittent dosing to continuous infusion. What's the patient population? Which are the patients who would really benefit the most? This trial included, of course, only ICU patients. Do you guys suggest that there should be even subpopulations within that group, or are there other populations?
So look, I think the kind of evidence-based way to answer the question in my mind is that I think beta-lactam infusion should be used for the patients who would have been enrolled in BLING-3. That is critically ill adults who are being treated with either piperacillin, tazobactam, or meropenem, full stop. Now, there probably are subgroups within them that are more likely to benefit. That wasn't shown within the BLING-3 trial, but I think that the subgroup that's the most interesting to me is those with organisms with higher MICs, in the higher MIC range, such as most Pseudomonas isolates, for example. In theory, I think they're more likely to benefit, but we don't actually have those data. One thing I will say, because there's been a lot of discussion about this, is what about non-ICU patients out on the wards? And I actually think it's important not to extend these trial findings to them. They don't have severe sepsis. They don't have the altered volume of distribution and supranormal GFR and all those sorts of things. And it's pretty easy to do it in intensive care. Patients have central lines with 4 lumens and all of that. It's not as easy to do it on hospital wards.
I'm going to disagree a little bit, Josh, and sort of take things a little bit laterally. All those patients we see down at the hospital entrance having a smoke, all having brought along their IV poles. Imagine a world where instead of bringing along your IV poles, you have like a syringe driver, like we do with outpatient antibiotics, and encourage patients' mobility who are out of the ICU on the wards or the floors, and be able to have the best of both worlds. If there's an advantage of continuous infusion of beta-lactams, they can potentially get that, but also get the benefit of not being tied down to an IV pole and that sort of hospital stay. I have to say, my take-home message is all beta-lactam antibiotics all the time.
I actually
Ooh.
agree. Okay, a couple things wearing my operations hat here. So I would go so far as to say continuous infusions are operationally sound and cost effective and time saving for the organization at large. Here's why. So a pharmacy technician that has to compound 4 to 5 doses or 4 to 6 doses of oxys, if you're giving something every 4 hours, I have to compound, or we call it affectionately, we call it pop, if you just take a drug vial and a bag and pop it together, or you have to take the drug and inject it into the bag. I have to have a technician, which, by the way, we have a 40% vacancy rate in pharmacy technicians because it's challenging to recruit into some of these positions. Across the globe, you may not have this. Nurses may do this compounding. Nursing time is unbelievably valuable. So whoever's making the IV bag, they either have to do it once or 6 times. No brainer, easier to do it once. Then for the nurse to actually hang and administer the bag, they have to spike it, they have to program the pump, they have to run the line, they have to attach it to the patient, they have to do all this. That takes like 15 to 20 minutes for every IV dose. If you do that once a day, you have given that, instead of 6 times a day, you have given the nurse an hour plus time back to do other patient related tasks. So operationally, continuous infusions are superior. Overall, I think it's not as challenging as people think it is.
Josh.
Can I do a brief pushback on the pushback? Agree central lines would be ideal, but we're not going to put a central line in every acute medical patient. I did a ward round this morning. I'm on general medicine service at the moment. Of the 20 patients I saw, probably 5 of those patients, I had to spend time silencing the alarm on their IV fluids or IV infusions because every time they bend their arm and move around, it's obstructed. I just don't think it's practical giving continuous infusions in peripheral cannulae. Those patients, while they're smoking downstairs, would be not sure what to do because their syringe pump would be alarming, unless they have a central line, in which case it would be okay.
That is true. Peripheral is hard in the hospital.
So I think we are all actually watching history happen. I hate to sound hyperbolic, but I really do feel like this is a huge paradigm shift. Some people have known about this for 30 years, like Jeff Lippman, but I think now we are witnessing what will be a huge, huge change. It's going to start in the ICUs. Let's see what happens if it goes outside the ICUs, like David would say, all beta-lactams all the time. I think it is, David. Yeah, ultimately, we could talk another hour about all the facets of the trial, the meta-analysis published in the same issue of JAMA, and the practicalities of actually implementing continuous beta-lactam infusion. The good news is on August 2nd, we will be able to download a Breakpoints podcast interview between Erin and Jason Roberts, who is the ID pharmacist on the BLING-3 trial and the senior author of the meta-analysis published in that same issue of JAMA. So do not miss that because you will feel smarter after you listen to it, as I always do after every Breakpoint episode. So Erin, thanks for being my co-host on this one.
Thank you. Your support means the world. Thank you on behalf of the Breakpoints team too, and love being on the ESCMID team now. So we're interviewing Jason next week. Actually, that episode will come out about a month later. We're going to get a lot more into the PK/PD. I kind of already went off about my pharmacy logistics, but we're going to talk a little bit more about that. So, okay, if you believe the data, now how do you operationalize it and implement it? A little bit more of that. So stay tuned. We're excited to continue this conversation.
And Josh, David, do you guys have any last words, last messages?
So I went from a skeptic before the study. Like I didn't really want to believe it. I had my, is PKPD really that everything? So I've gone from a skeptic to a real believer. And I think that's the value of clinical trials, particularly big clinical trials.
I was thinking that. I was like, the guy that said he wouldn't have even done the trial is now like, give every human in every location a continuous beta-lactam. I was like, this has been a roller coaster of an hour. So
Yeah. I'm a little bit less enthusiastic than David, but I am convinced by the data. I think beta-lactam continuous infusions save lives in critically ill patients, but the effect size is small. So if it's possible to do it, it's practical to do it, you should do it. And I'm talking about patients who are critically ill in ICU. It's still an open question in my mind, other settings.
Thank you very much. And thank you for listening to Communicable, the CMI Comms podcast. This episode was hosted by Erin McCreary and Angela Huttner, editors at CMI Comms, ESCMID's open access journal. It was peer-reviewed by Dr. Mia Lidén. theme music was composed and conducted by Joseph McDade. You can subscribe to Communicable on Spotify, Apple, YouTube, wherever you get your podcasts, or you can find it on ESCMID's website for the CMI Comms Journal. Thanks for listening and helping CMI Comms and ESCMID move the conversation in ID and clinical microbiology further along.